Hey everyone, it's Mr. Sinti, and today's podcast is on lipids. Lipids are one of four very critical macromolecules that are involved in biological organisms. And what's interesting about lipids right out of the gate is that they don't really commit themselves to being polymers. So they're not true polymers in a sense, but they are large. And one of the things that I want to say about lipids right out of the gate is that one of the unifying principles between lipids and the types of lipids is that they all share this sort of common thread of being hydrophobic. In other words, having no affinity to bond with water. Now, there are a few exceptions to that, meaning a phospholipid has a portion of the molecule is slightly polar, and so that's kind of interesting as well. So they all have no affinity for water for the most part, and that's because they're dominated by nonpolar covalent bonds. And so there's a lot of carbon-hydrogen, carbon-carbon bonds. There's a little bit of oxygen, and so if I were to just sort of do that and say that there's a lot of carbon, there's a lot of hydrogen, and there's not so much oxygen. And then I mentioned the phospholipid containing a little bit of the element phosphorus. And so what's interesting about this, won't get too far into it, but it's suggestive of the fact that these carbon-hydrogen bonds contain a lot of potential energy, and that lies the secret to one of the main functions of lipids, which is that it is a fabulous storage of energy. And as you can see in this picture below of all these cells, these are a group of cells collectively known as a tissue. It's a type of connective tissue called adipose tissue. And adipose tissue contains lipids inside its vacuoles inside here, and this is what stores energy. It could also help to insulate us, to keep us warm, and cushion us from traumatic shock from the outside. It surrounds a lot of our organs, and so it's rather important. So the thing about it is, sometimes people, I'll just come right out and mention this forward with this, is that some people think that lipids are mostly fat and that fat is a bad thing. But the truth is there's great diversity in lipids in both form and in function. And so I hope this podcast illuminates that and gets over that misconception that fat is bad. Because fat is something that's essential, and clearly too much of it is harmful. So although fats are not strictly polymers, as I said before, they're very large, and they're assembled by some smaller subunits, not altogether that small, but they're assembled through dehydration synthesis. And so fat is composed of two kinds of molecules, and that is a glycerol. And what's interesting about glycerol is that it's in it of itself, that particular molecule has 3 carbons like this, and it's an alcohol. And it's somewhat curious because, as being the alcohol that glycerol is that in it of itself it's very polar because of these polar covalent bonds. But when it's united with a fatty acid, it becomes a completely nonpolar molecule, or hydrophobic. And so let's consider that. Here's that glycerol that I just drew in gray, and here are the alcohol groups or hydroxyls coming off of it. The second component of a fat is something called a fatty acid. And simply what a fatty acid is, a hydrocarbon chain. You can see there's many carbons here. Now, if you notice, all of these are CH2 in the center, and then the C terminus is CH3. So you could do something like CH2 like that, N, because fatty acids can vary in length. And then there's the CH3 at the very end. And then over here, this is the fatty part, and the acid part is right here. That functional group, you might recognize it. It's C double bond oxygen at the end of the molecule connected to a hydroxyl. So collectively, this is known as a carboxylic acid. A carboxylic acid right there, because it has a tendency to donate protons in certain pHs. So when the carboxylic acid of a fatty acid unites with a glycerol, water is liberated. And as it turns out, it requires 3 fatty acids to complete a fat. Now, these fatty acids can vary in length. They're often about 16 to 18 carbons in length. And they can be symbolized. So I drew a shorthand before of that, but I can also go like this. In other words, everywhere you see a point, there's a carbon. So I can draw it like that, and then there's your carboxylic acid at the very end. Right there, you could do it that way as well. And so what's interesting is when the 3 fatty acids connect through dehydration synthesis to the glycerol, it forms an ester linkage, which is what this is right here. So here's a triglyceride. Now all of this is nonpolar, these carbon-hydrogen bonds, and so it creates the triglyceride, which is very hydrophobic. And so in order to animate that, I thought you might like to see this. You can see here's the 3 fatty acids, and when they unite with the glycerol, they come together and each of them produce water. And so as a result of it, it's a glycerol plus 3 fatty acids makes a triglyceride and 3 waters. And so thus it's a condensation reaction, and the triglyceride is formed. It's also called a dehydration synthesis reaction. Now, why would you do that? Why would cells produce triglycerides? Well, the main function of a fat is the storage of energy. There's a lot of potential energy in these bonds right over here in the fatty acids, and so it's tremendous. And so again, let me do this. Let me come back here and get my ability to draw, which is good. Come over here, and if I do this, here's my glycerol, like that. And then there's my alcohol group coming off of it. Here's my glycerol, like that. There's my hydrogens coming on over here. So here's a glycerol. So it takes 1 glycerol and 3 fatty acids. I'm only going to draw 1 of the fatty acids. And what I could do, I can go even lazier still. I could say something like, there's an R, which is the fatty part, and here is my carboxylic acid coming off like this. Here's my fatty acid. And so what's happening, I just want to illustrate this, what's happening is the removal of water right there, and then the ester bond is formed between the fatty acid and the glycerol. And so what's really interesting is these fatty acids can vary in length. They could be a little shorter, they could be a little longer, but nevertheless, what's fascinating about them is that if there's single bonds between the carbons, they're considered to be saturated because they contain all of the hydrogens. If there's a double bond present, you may recall from a previous podcast, a double bond suggests a unsaturated bond, which means there's only 1 hydrogen coming off of those 2 carbons in the double bond. And it can either be in a cis, which is up, or down, or trans, up-down. And so you can get some different isomers. And so what we say is that a fat is truly saturated, meaning that there's single bonds in the fatty acid tails right here. And a classic example of a saturated fatty acid is stearic acid coming from a steer or a cow. And so what's interesting about this is that the fat is considered, like butter represents this, is considered to be solid at room temperature. And the fact that it is a solid at room temperature means that it's fairly difficult to melt. And that is because the fatty acid tails can pack very closely together, and van der Waals forces or London dispersion forces can occur between the fatty acids, therefore maintaining a higher melting point at room temperature, and so therefore solid. And so saturated fats are infamously associated with cardiovascular disease. We could certainly want to reduce this in our diet. Study after study has shown that diets that are found with a lot of saturated fat can lead to cardiovascular disease and arterial sclerosis. So we want to kind of slow down the saturated fat. the thing about it is, truthfully, I think most people enjoy the taste of saturated fats, and I think my speculation to this is it may not be factual, but I honestly believe that one of the reasons that that we intrinsically enjoy fatty acids, especially the saturated ones, are because of the fact they are very caloric. And evolutionarily speaking, it's advantageous to consume something that has a high calorie content. It's only most recently that we have so much food that this can become a problem. And so it's encouraged to eat fatty foods because they taste great. So it's sort of a natural selection kind of thing. Now, if I mentioned this before, if there's a double bond that occurs in the fatty acid, say there was one, it's considered to be monounsaturated fatty acid, and it puts a kink or a bend in the molecule. And the kink in the molecule, especially when considering the triglyceride, let me see if I can go with that. If you put a kink in the molecule like this, it's coming like this, and you put a large bend in it, it sort of puts it away from the other fatty acid, and it prevents van der Waals forces from occurring because the distance is too great. And so many unsaturated bonds are called polyunsaturated. So if you had polyunsaturated fatty acids, that means there's many double bonds, and it becomes an oil. So triglycerides and triglycerides can either be saturated fats or unsaturated fats. And if it's solid at room temperature, it's considered to be a fat. And if it's a liquid, it's considered to be a oil. And that's because of the kinky nature of the double bond preventing the van der Waals forces. And so fatty acids could vary in length. Number of carbons determines that, or the degree of saturation. You can have unsaturated and saturated fatty acids. And again, I was mentioning this, that plants can have, fish can have oils which are liquid at room temperature, and the double bonds prevent the molecules from tightly packaging. And so I mentioned before, the saturated fat, of course, can be turned into a liquid if heat is applied. But they're solid at room temperature. And the thing is, I won't go into the detail of it, but saturated fats can be a real problem in the blood. And this is inside of an artery. All arteries are important, but geez, the ones that are supplying the heart with blood, which are coronary arteries, these are even more critical. And even the arteries in the brain are critical as well, because if you get a blockage here or plaque, you can narrow the blood flow and therefore cut off the red blood cells entirely. And if the heart, which is a really active pump, is deprived of oxygen, the muscle itself could die, and that's called a heart attack. And so this could lead to death. And so you really want to slow down that intake of saturated fat. But again, on a positive note, fat is a great storage of energy, and it's literally double. It's 9 kilocalories versus 4 kilocalories in carbohydrates. So it's about twice the amount of energy than does a carbohydrate contain in protein. And this is useful. And so in other words, if you're deprived of food and you're eating fats, you're likely to survive. And so another pro point about fat, other than it's a great storage of energy, is I was mentioning that because of the fact that it's hydrophobic, it means that the triglyceride isn't really adhering to a tremendous amount of water. If you're a polysaccharide, like glycogen in an animal, that's really heavy because all of those glucoses have water clinging all over it. It's sort of like a beach towel that's thrown into a swimming pool. It's really, really, really heavy. But fat, paradoxically speaking, ironically speaking, is a lightweight storage of energy. It's lightweight. And it also helps, again, insulate us. It keeps the heat in, and it also it cushions us. And I know you may keep the heat in is maybe not the top of your list of priorities, but it is if you're living in the ocean. If you're a big elephant seal, you got a lot of blubber. If you're if you're a whale, you have a lot of blubber, and blubber is fat, and that helps for insulation. And it's found right below the skin. No surprise there. So it's subcutaneous, below the skin, hypodermis. And so marine mammals have a lot of this. Now, in addition to triglycerides, that was the first type of lipid, we have phospholipids. Now, these phospholipids are the really cool ones, because they're the ones that contain a little bit of phosphorus, as I was mentioning before. And not only that, is that they sort of break the stereotypical all hydrophobic, because a portion of a phospholipid is polar and the other portion is nonpolar. And so what we've got going here with the phospholipid is it's made up of a glycerol, just like a triglyceride. It has two fatty acid tails, but instead of the third fatty acid tail, it has this phosphate functional group right over here. Let me just draw it in as a circle. And so it has a circle which is negatively charged, because the phosphate is negatively charged. Here's your glycerol, and it has two fatty acid tails. And sometimes it's simply drawn like this, a head and two tails coming down like this. This is the polar area and the nonpolar area. And so what's interesting is the phosphate group carries a negative charge, but the fatty acids are nonpolar. So they're similar to a triglyceride, but different in a significant way, in that this head area is hydrophilic and the tail areas are hydrophobic or nonpolar. What's interesting about a phospholipid, many things, but often if there's a double bond, take a look here, there's a kink, and this is what we mean by this. And so in other words, there's van der Waals forces that can occur here, but then this part is too far away. So the tails are a perfect little blend of like viscosity, a little bit of thickness, but not solid, kind of like thick oil, if you will, a phospholipid in terms of this. And so that is important in the structure of the cell membrane, which we'll get into the details. I'll allude to it right now, but we'll get into the details of it in a separate video. If you toss a phospholipid into water, not to say that, why would you do that, but just saying, if you were to toss a phospholipid into water, it might land on its head, because the polar head interacts with the water, and so this is what you'd get. And also, if you threw these phospholipids in, they might form a little ring like this called a micelle, shielding the innards from the water, because the heads are hydrophobic. And so, if I mentioned the cell membrane is made up of phospholipids, but this doesn't make sense, because if the cell membrane is made of phospholipids, the inside of the cell is water as well. And so the way in which that works, you may already know this somewhere in your past, is that the cell membrane itself is a phospholipid bilayer. Ooh. So there's polar heads on the outside and polar heads on the inside, with the tails pointing on the inside of the membrane, because this is in contact with water on the outside and the cytoplasma on the inside. Cyto means cell, plasma means liquid. So the liquid on the inside is in contact with the heads. The extracellular aqueous solution is also in contact with the heads. So that's kind of interesting. So the phospholipid is the major component of the cell membrane. There's other things embedded in the cell membrane as well, but it's the main ingredient. And so here's a picture of the phospholipid bilayer. So heads are on the outside and on the inside. And when I say other things, I mean there's some proteins and even another lipid that we're going to mention in a moment called cholesterol. And there's even some sugars on the outside of a cell membrane. So here's a cartoon drawing of the cell featuring the phospholipid bilayer right here. The dark red is our polar heads of phosphate groups. What's not shown is the fact that there's proteins and there's cholesterol and there's sugars also involved. But the main ingredient of the plasma membrane and also some other organelles are phospholipids. Extremely important phospholipids, just saying. Now, the third type of lipid that I want to introduce in this video are steroids. Steroids are lipids. Just as a triglyceride, just as a phospholipid, steroids are lipids. Now, one of the main lipids, the most common one, is cholesterol. So cholesterol, here's a diagram of cholesterol, and it doesn't quite look I mentioned before that these are not polymers, and it doesn't really look like the triglyceride. It's a little bit different. It's got 4 rings of carbon, and this is actually cholesterol here. And this 4-ring base is fundamental to all steroids. Cholesterol is one type of steroid, but there are others. And you might know steroids are associated with hormones. So, many hormones are steroids. Not all hormones, which are chemical messengers, are steroids. They're called, funny, non-steroid hormones. They can be protein or little peptides. But steroid hormones basically look like this. And I say basically look like this. They have the 4 rings of carbon. And depending on what functional groups are attached to these 4 rings, you can get a myriad of different hormones. You get estrogen, you can get testosterone, aldosterone, all kinds of different hormones based on the functional groups. Now, this is essential. We use hormones all the time. They're not bad. We use them all the time. The cells produce them, and they're chemical messengers, and they initiate a response in other target cells. And so they're a way in which cells communicate using these lipids. It's kind of interesting. Now, cholesterol in particular isn't used that way. It's used in the cell membrane to maintain membrane fluidity. I'll get into that in a moment. But I just wanted to point out that steroids, the name precedes this conversation. I know when you hear the word steroid, it's sort of a negative connotation comes up, just like fat, even though I hope I convinced you that fat is pretty good. Like, for example, our San Francisco Giants, world champions, 2 years, 2010, 2012, fabulous. Here's Melky Cabrera, and he was allegedly using steroids. And so what's that all about? Well, if you're injecting yourself with testosterone, for example, which is a steroid hormone, that could promote growth and strength of muscle. And so this kind of thing shouldn't be allowed because it's an advantage over other athletes. And so cholesterol is a steroid that's found embedded in the phospholipid bilayer of cell membranes. And notice it's this nonpolar area that embeds between the fatty acids. And what happens, and I'll talk about this in a later video, is that it prevents the close packaging of the fatty acid tails, and so therefore it reduces van der Waals forces between fatty acids, and so therefore it maintains membrane fluidity. And the membrane fluidity is critical for proper functioning if you're going to allow molecules to pass in and out and different sorts of things that cell membranes need to do. So cholesterol is rather important. Cells can alter and adjust the quantity of cholesterol in the membrane, which is kind of interesting. Now, if you don't consume cholesterol in your diet, the body will make it. It's so critical. The body will actually synthesize cholesterol in the liver. And so you can tell that it's essential. And there's a few fatty acids. I didn't get into it because I didn't want to take too much of a nutritional angle, but there's a few essential fatty acids that need to be consumed as well. So you don't want to be the hater on eating fats and cholesterol. And so the thing is, if you did eat a little bit too much cholesterol, a moderate amount, then the body could take that precursor like this right here and actually rip off a couple functional groups and turn it into things that it actually needs, like, for example, hormones. And so maybe you need some testosterone, and so you can take cholesterol, which is found in the egg yolk, and you can turn it into testosterone. That would be great for you if you needed that. And so again, you have to be careful because high amounts of cholesterol can lead to cardiovascular disease. So lipids are really cool. There are triglycerides, there's phospholipids, there's steroids, and I didn't even mention waxes, which are a really interesting type of lipid that's kind of simple looking. They're mainly fatty acids that have ester groups and some alcohol associated with them, those sorts of things. They could store energy. They're mostly hydrophobic. They can be insulators, and they can be for cushion, and they can be communication in terms of hormones, or maintain membrane fluidity in the case of cholesterol. So extreme diverse form and function with these lipids. So I hope you enjoyed this brief look at lipids. Thanks for watching.